Hello, my name is Steve Horvath. I'm an aging researcher. By training, I'm a bioinformatician, but lately I'm trying to understand how to measure aging in different mammalian tissues. And I will talk to you today about epigenetic clocks and more generally DNA methylation studies of aging in mammals. Um, My entire talk is based on cytosine methylation. Cytosine methylation plays a critical role in the development of vertebrates. A zygote could never differentiate into other cells without methylation. So methylation is critical in vertebrates, not in invertebrates, but vertebrates. Um, My data are typically being generated on an array platform, the so-called Infinium array from Illumina, and I will be using two types of platforms in this talk. One is the human EPIC chip, which measures over 860,000 cytosines in the genome, and the other platform is a custom methylation array that measures 37,000 cytosines in highly conserved stretches of DNA. This custom array is well suited for comparative studies in mammals, as I will describe. And these arrays provide methylation levels, which are really numbers between 0 and 1. The mammalian methylation array is described in a preprint by Adriana Arneson and Jason Ernst. And I will briefly mention some ideas behind the design of the mammalian array. The top row shows you an oligonucleotide sequence, where on the right-hand side you see that there's a C and G pair, the cytosine guanine pair in humans. But if we want to use the same oligonucleotide for other species, we need to build in tolerance to mutations. For example, in chimps, you see the yellow letter C is different from the original nucleotide sequence. And in general, as we look at many different mammalian species, there will be mutations. And now the mammalian array can tolerate a few of these mutations per nucleotide sequence. And therefore, we can measure CG, cytosine methylation, in many different species. Here I show you how many cytosines apply to different phylogenetic orders. The top row shows you primates, and typically the mammalian array profiles at least 33,000 cytosines in primate species. In rodents, you see we have about 28,000, in bats even more. And however, as we go to evolutionarily very distant species, these monotremata, for example, we would have fewer than 15,000 cytosines. But interestingly, 70% of the CpGs map to orthologous genes in humans, and that's very exciting, because imagine you study cytosines in a mouse or a bat, then typically these cytosines are located near the same gene as in humans. And that highlights really the virtue of the mammalian array. It allows you to look at genes and pathways that are conserved between many different mammalian species. To date, we have generated over 10,000 tissues from 175 mammalian species. Here I highlight in red and in blue different phylogenetic orders for which we have animals. And really many cetaceans, whales, deer, practically all animal species that are being used in agricultural settings, bats, and so on. I will now describe epigenetic clocks for mammals. Epigenetic clocks are meant to measure the chronological ages of different tissues and body parts, not just in humans, but of course also in different mammalian species. I'll start out with bats. We just published a paper whose title says it all. So we show that one can predict age in bats, but also methylation provides insights into the exceptional longevity of bats, because some bat species can live for more than 40 years. So here I show you in the scatter plot our epigenetic clock. On the x-axis, we report chronologic age of these different bats. And you see we had samples from bats that were about 23 years old, so pretty old bat samples. Conversely, we also had very young bats, newborns in essence. And we had wing punches from these bats, tiny amounts. And in the panel, I show you the Latin names of 26 different bat species. It turns out there are over 1,000 different bat species, but at least these 26 bat species are representative, of course, of many of them. And what you see is a very high correlation between the estimated value of age on the y-axis and chronologic age. And the dots are labeled by the bat species. So it's really remarkable that you can have one clock, one epigenetic age estimator, one mathematical formula that allows you to measure the ages of all of these different bat species. And we're quite confident that this clock for bats also generalizes to bat species that were not part of our training set. By the way, all the scatter plots I show you are based on cross-validation or independent data. So these are unbiased assessments of accuracy. We also have highly accurate epigenetic clocks for dogs. These clocks apply to all dog breeds. Ideally, we like blood samples from dogs. And here we profiled 51 different pure dog breeds in collaboration with Elaine Ostrander. And so our clock is remarkably accurate, as you can tell. And the different colors correspond to different dog breeds. We also have very accurate clocks for an amazing animal, which is the naked mole rat. Why is it amazing? Because this animal has what is dubbed negligible senescence. It doesn't seem to age. Although this animal is roughly the size of a rat, it can live for up to 37 years, so remarkable longevity. However, as you can see from these different panels that describe pan-tissue clocks, meaning a clock that applies to all tissues from the naked mole rat. Panel B shows you an epigenetic clock for blood samples from the naked mole rat. Panel C for liver. Panel E for skin. So interestingly, we can still build very accurate epigenetic measures of age in the naked mole rat, even though on a phenotypic level, it seems to have negligible senescence. Moving to rats. Rats, of course, live far shorter lives than the naked mole rat, let's say less than 4 years. And here I show you a pan-tissue clock for rats. So we have very accurate aging measures for rats. If you carefully look at the x-axis, we restricted the analysis to animals that were less than 2.5 years old. And the middle shows you a clock for brain sample, the right-hand side a clock for blood sample. So we have very accurate clocks for rats. However, you can do something that seems to be outlandish. You can actually build a clock that applies both to humans and rats at the same time. So it's one formula that allows you to estimate the ages based on highly conserved cytosines in humans and rats. And we actually have two such clocks. One is called the human rat clock for absolute age or chronological age. It really estimates just calendar age. But we also have another human rat clock for what we call relative age. Relative age is defined as the ratio between age and the maximum lifespan of the species. So by definition, the relative age is a number between 0 and 1. 0 is the age at birth, 1 means maximum lifespan. And why do we do it? Because relative age allows us to align these species and provide a biologically more meaningful comparison between species when it comes to different aging rates. The right-hand side shows you how our human rat clock applies to rat samples, so the correlation is still 0.93. It's remarkably accurate. The left panel shows you the mixture of human samples colored in green and the orange samples, which are the rat samples. So you see that this clock really measures aging in these animals of very different lifespans. One could, of course, become more ambitious, which is, well, can one build a universal mammalian clock that measures age in all mammalian species? So we assembled a large collection of species in order to build such universal mammalian clocks. And here I show you some results from our preprint on bioRxiv. We've built actually several universal clocks. Panel A and B describe universal clocks for placental mammals. Panel C on the right shows you a universal clock for marsupials. I distinguish eutherians or placental mammals from marsupials because they are sequence divergences. But long story short, we have remarkably accurate universal clock. So each dot here is a tissue sample from a different species. The dots are labeled by the species as outlined in the previous legend. So here we have different species. And I think you can appreciate this very high accuracy, in these nearly 10,000 tissue samples. But beyond epigenetic clocks, you can ask a simpler question. Can you find cytosines that correlate with age in many different mammalian species? So in other words, one can do an epigenome-wide association study of age across these, I think it was 128 species at the time. And so this analysis is very insightful because we do find cytosines near genes that have very high correlations with age in all species or most species. So there's one cytosine near the gene LHFPL4, and this cytosine has a very strong correlation with age in pigs, as you see in panel B, high correlation with a deer mouse, panel C, and primates, panel D, which is a particular monkey, the vervet monkey. I want to come to another question, which some people have called the central question of biology, which is, why do similar species, such as mammals, have markedly different maximum lifespans? Right? Why do humans live up to 122 years? Why do mice live for less than 5 years? Why do some bats live for over 40 years? And there's really a substantial literature on that topic. I mean, all goes back to an essay from Aristotle. And people have found wonderful answers in ecological studies. In brief, animals that evade predators often live substantially longer than animals that have lots of predators. However, the molecular reasons really require more detailed analysis. And my angle to that question is, of course, to look at methylation profiles and epigenetics. And here I show you one cytosine which is highly correlated with the maximum lifespan across species. So the y-axis shows you the logarithm of the maximum lifespan. The x-axis is just a single cytosine. And you notice a very strong negative correlation. Each dot is a different species. That's one cytosine. Question is, what if you combine many cytosines? Can you build a more accurate estimate of maximum lifespan? And the answer is yes. So Cesar Lee in my lab has developed a multivariate predictor of maximum lifespan based on a penalized regression model. So here each dot is a different mammalian species, and the correlation is 0.88. So these estimators work remarkably well. I want to now come to my favorite mammal, which is, of course, humans. I'm motivated by identifying and validating anti-aging interventions in humans. And I want to describe the so-called human pan-tissue epigenetic clock, which was published in 2013. This which applies to all human tissues and cell types that contain DNA. Now, in general, epigenetic clocks allow you to create a curve here indicated in blue or line that indicates the expected age. But people whose methylation age estimate lie above this line, they are said to exhibit positive epigenetic age acceleration, and individuals who lie below the lines exhibit negative age acceleration. And epidemiological studies have shown that people with positive epigenetic age acceleration are at a slightly higher risk of various age-related conditions. But it really depends on the clock. For example, the pan-tissue clock is not the best mortality risk predictor. Rather, we use GrimAge that I will describe later. But I want to now show you a more detailed analysis of the pan-tissue clock. The pan-tissue clock allows you to measure age acceleration in many different tissues. For example, human adipose, human kidney, human liver, muscle, and so on. And you can ask the question, well, which age-related conditions are associated with age acceleration in these different human organs and tissues? And the striking thing for me was that hypertension really pops up. So the first row shows you that hypertension is associated with epigenetic age acceleration in adipose tissue, kidney, liver, muscle. Other conditions, diabetes is associated with age acceleration in the liver. Cardiac disease is associated with age acceleration in lung and adipose tissue, and so on. So these kinds of studies show us that many age-related conditions are actually specific to a given cell type or organ. I mention it because everybody studies epigenetics in blood tissue because blood is so readily available. But blood is not always informative, so you want to carefully think about what cell types should be profiled. We also evaluated the effect of sex on epigenetic age acceleration, and interestingly, or perhaps you expected it like everyone else, males show epigenetic age acceleration compared to females in really most tissues that we looked at. Certainly in blood and also brain and spleen and so on. The effects are relatively weak in blood, so males are maybe 1 or 2 years older than females in blood. In other organs, the effect may be more pronounced. I now want to come to what we call the second generation epigenetic clocks for humans. These clocks should not be interpreted as age estimators, but rather as mortality risk estimators. There are 2 clocks that are widely used in the literature, the so-called PhenoAge clock and the GrimAge clock. The GrimAge clock is the best mortality risk predictor. It really predicts lifespan when you use retrospective studies, so blood samples collected, let's say in the 1990s, then you have follow-up information on these people, and I'm sure if you have a reasonably sized data set, maybe 300 people, you can validate these claims, that GrimAge predicts lifespan. And how strong are the effects? Well, if you're in the top 5% of the fastest agers according to GrimAge, then your mortality risk is 2 times that of the average person. So, and I will flesh that out. Mortality risk means hazard ratio of death from a Cox model. So the GrimAge clock was published by R.K. Liu, and here I show you the results from a large-scale evaluation of that biomarker. So we evaluated age acceleration, meaning we, of course, adjust for chronologic age and sex in the analysis. And we find that epigenetic age acceleration based on GrimAge is very strongly associated with time to death. The p-value is 10 to the minus 75. Now, let's carefully look at the hazard ratio from this Cox model. It's 1.1, which doesn't seem like a lot, but 1.1 is the hazard ratio associated with 1 year of age acceleration. And however, in the bottom, I show you the calculation that 8 years of epigenetic age acceleration for a person would be associated with a hazard ratio 2.14, so which is a strong effect. About 5% of the population have an epigenetic age acceleration of 8 years or more. Conversely, if you're lucky enough that your epigenetic age acceleration is, let's say, 8 years younger than expected, then your hazard ratio of death would be half that of the average person. Here I show you a study of healthy lifestyle in the Women's Health Initiative. For these women, we had blood measures of vegetable intake, more precisely blood measures of carotenoid levels. And what we see is that, yes, women, postmenopausal women who eat vegetables, they age more slowly according to GrimAge. The green column shows you the p-value, so there's no question that these are very significant associations. At the same time, these associations are based on at least 2,200 women, so these are large sample sizes. At the bottom panel, you see that educational level is weakly but statistically significantly associated with slower epigenetic aging according to the GrimAge clock. Exercise also And of course, smoking is very strongly associated with positive epigenetic age acceleration according to GrimAge. And that's a little bit by design because the GrimAge clock involves cytosines that change a lot with smoking. In general, anything related to metabolic syndrome, obesity, insulin levels, and so on are associated with faster GrimAge. So to summarize that all, you want to lead a healthy lifestyle in order to slow your epigenetic aging clock. The next step are really to work on qualifying epigenetic biomarkers as surrogate endpoints for human anti-aging studies. We're not there yet because in order to qualify any biomarker as a surrogate endpoint, you really need to provide very detailed evidence that this is the case. What I can tell you is that GrimAge has been validated as a mortality risk estimator by a dozen papers, many by independent groups. We're currently carrying out longitudinal studies of GrimAge. I want to also mention we have published a large-scale GWAS of GrimAge, but also other epigenetic clocks, and we have polygenic risk scores for these epigenetic biomarkers, so you can download them for free. So if you have SNP markers, you can relate these polygenic risk scores to your favorite age-related condition. We also carry out analytical validation studies because we want to show that our biomarkers have low retest variability. And GrimAge stands out again by low variability. So GrimAge is our best predictor of time to death, time to heart disease, time to cancer. It also relates to early age at menopause. GrimAge uses a methylation-based estimator of smoking pack years, which interestingly outperforms self-reported smoking pack years in predicting lifespan. So in conclusion, we have managed to build epigenetic clocks for each mammalian species that we analyzed. We have universal clocks that apply to all species. Human clocks differ in predictive accuracy for mortality risk. My pan-tissue clock is a weak predictor of mortality risk. GrimAge is a strong predictor of mortality risk. DNA methylation strongly relates to differences in maximum lifespan across different species. And in humans, methylation data are predictive of mortality risk even after adjusting for standard risk factors. Methylation age will not replace any standard clinical measure that are being used as part of your physical. It will not replace blood pressure or lipid levels or glucose measures, so it complements them but will not replace them. And as you design your own study, you want to think carefully about the tissue or cell type that you want to study. As I've shown you, some conditions are associated with age acceleration in adipose tissue, but not in blood or in muscle tissue and so on. So you can generate methylation data from blood, saliva, buccal swabs, even urine contains enough DNA, and so you can think about it. I want to end by thanking my many collaborators. I mentioned several people already, and I'll stop here. Thank you so much.